Hello, and welcome to our lecture on applications of DNA-based technologies. This is Chapter 9 in our textbook. In biotechnology, we are using those concepts and mechanisms that we learned about in DNA replication, transcription, translation, horizontal gene exchange, all of the different genetic properties of these microorganisms and some of their mechanisms, and we use them to our advantage in the laboratory so that we can gain a greater understanding of how DNA works or where the location of a gene is. Maybe we want to repair a gene or knock out a gene so we can see what the product result is in that cell. So there's lots of different reasons that we might manipulate or work with the genetics of an organism in order to really understand on a molecular level how it works. So there are some fundamental tools that are used in biotechnology, and we are looking at the actual tools themselves. What these tools can be used for is almost endless. So we have combinations. Some of these tools will be used together, but it's kind of like I always describe microbiology as having a big toolbox. And whereas microscopes are one tool, we have different types of microscopes in our toolbox, we also have different types of biotechnology in our toolbox. And so we can take these tools and use them together individually for different purposes to look at microbial genetics and eukaryotic genetics, for that matter, in a multitude of different ways. Another thing I'd like to just briefly mention to you is that this particular lecture is going to be very video heavy. I will be showing you quite a few videos because these tools, although I can describe them and show you pictures and we can talk about them, sometimes it's really difficult to really grasp or kind of wrap your head around what these tools are actually doing, especially when first learning them. So I did include a lot of videos in here to give you a good visual foundation of what these different tools look like in action. So the first are restriction enzymes. We'll look at gel electrophoresis, a very new technology just in maybe the past 5 to 10 years, which is the CRISPR-Cas9. This really has some really amazing promises in the future. This CRISPR system is really exciting. We have sequencing, PCR, which is known as polymerase chain reaction, probe technologies, and genetic engineering. So restriction enzymes, we're going to start with. Restriction enzymes are bacterial enzymes, and they are produced by bacteria to protect themselves from viral infection. There is a set of viruses, a class of viruses called bacteriophages that infect bacteria. And when they attach to bacteria and infect them, they will inject their genetic material in DNA form. So viral DNA will be present inside of the bacterial cell, and the bacterial cell does not want this viral infection because it can induce mutations and it takes over the bacterial cell, and eventually the cell will die from it. So the bacterial cell will try to protect itself, and it will use these restriction enzymes to do this. The restriction enzymes will look for sequences in the viral DNA that are called palindromes or palindromic sequences. These palindromic sequences are sequences in DNA that are spelled the same 5' to 3' as they are backwards on the 3 to 5'. So what I mean by that is, let me get a little pointer here. I'm going to get our pointer, and we're going to go back. So what I mean by that is here I have my sequence GAATTC, and my opposite, my complement strand backwards is GAATTC. So this right here, this whole thing right here is a palindromic sequence. And EcoR1, this very particular restriction enzyme, recognizes that sequence. Now, what makes restriction enzymes incredibly useful is when they cut, they don't cut out their sequence. They just find a region within that sequence and they just cut the sugar phosphate backbone. This means that where you see this red dotted line, the only thing holding these two strands of DNA together are a couple of hydrogen bonds, which are very weak individually. So these two strands will fall apart from each other, and now I have these two this is double-stranded here in blue, but where the restriction enzyme cut, I have some single strands. I have a couple of bases that are overhanging that are single-stranded. Now this is really important. These are referred to as sticky ends. And these sticky ends are used to create new recombinant DNA molecules. So if I have a restriction enzyme and I cut a plasmid, a bacterial plasmid with this restriction enzyme, where the plasmid is cut and now open is called sticky ends. If I have DNA from another organism, for example, over here on the right, we have DNA source number 1 and DNA source number 2. If I cut both sources of DNA from two different organisms, they're two different cells, two different organisms. If I cut them both with the same restriction enzyme, I'm left with these sticky ends that will be complementary to each other. The reason for this, I used the same restriction enzyme, which left me the same palindromic sequence in both organisms. Where these sticky ends match, I now end up with two new recombinant DNA molecules, because the base pairing will cause the sticky ends to base pair to each other. DNA ligase can be added to the mix to repair the sugar phosphate backbone, and now I have a new DNA molecule, or a new recombinant DNA, where I have DNA from two different sources. So I'm going to show you a quick video here. This is a short video that kind of tries to show you in real time and in real structure what a restriction enzyme function looks like. So here we have DNA in its helical form, and this restriction enzyme attaches, and it reads the DNA, and it's reading along the DNA until it finds its sequence, and it cuts it, and there they're showing you the sticky ends. Now here we have DNA coming in from another source. Base pairing occurs. DNA ligase repairs the sugar phosphate backbone, and we now have a recombinant plasmid. And that's how restriction enzymes and recombinant DNA is made. So here we'll show you another video. It's just a little bit longer, but it's the same idea, and it does talk about the phages.
Using genetic engineering, scientists can create cells that produce proteins they wouldn't ordinarily produce. For example, circular pieces of DNA, called plasmids, found inside bacterial cells, can be engineered to make such proteins as human insulin by inserting the gene that codes for that protein. Special enzymes called restriction enzymes, which recognize, bind to, and cut specific DNA sequences, are used to open up a plasmid in preparation for the insertion of a gene. Some restriction enzymes leave the DNA with uneven ends, also known as sticky ends, shown here. If the gene we wish to insert and the plasmid are both cut with the same enzyme, these sticky ends will be complementary, meaning they will readily bond with one another. In a follow-up reaction, gene fragments, cut plasmids, and an enzyme that connects DNA fragments are mixed together. This so-called ligase enzyme attaches the sticky ends in the solution to produce circular plasmids that now carry the gene that codes for the desired protein. Note that the efficiency of recombinant techniques is not 100% and may lead to plasmids with no inserts or multiple inserts.
So in a later lecture, we will come back to these plasmids, and we will look at ways that we can differentiate between bacteria that have plasmids with no inserts, plasmids with the correct insert, and plasmids with multiple inserts. See you in the next lecture.